We continue with cytokinesis. Cytokinesis is the physical process of cell division, which divides the cytoplasm of a parental cell into two daughter cells. It occurs concurrently with two types of nuclear division called mitosis and meiosis. In animal cell, cytokinesis occurs by a cleavage process. It forms the cleavage furrow, which is the shallow groove in the cell surface at the old metaphase plate. It forms with the help of contractile ring of actin and myosin. On the cytoplasmic side of the furrow is a contractile ring of actin microfilaments associated with molecules of protein myosin. Then, the contraction of the ring deepens the cleavage furrow, until the parent cell pinched into two new cells, each with its own nucleus and its own share of cytosol, or organelles, and other subcellular structures. Let's watch video of cytokinesis in animals.
Cytokinesis is the phase of the cell cycle during which the cytoplasm actually divides. It follows nuclear division and involves the splitting of the cell into two cells. In animal cells, cytokinesis is achieved by a constricting belt of actin filaments interacting with myosin. As the filaments slide past one another, the diameter of the belt decreases, pinching the cell and creating a cleavage furrow around the cell's circumference. Constriction proceeds, and the furrow deepens until it eventually slices all the way into the center of the cell. At this point, the cell divides in two.
Is cytokinesis in animal and plant cell same? No. Cytokinesis in plant cells, which have the cell walls, is markedly different. There is no cleavage furrow like animal cell. In plant cell, cytokinesis occurs by formation of cell plate. Instead, during telophase, vesicles contains the cell wall materials derived from the Golgi apparatus move along microtubules to the middle of the cell. Then, they fuse to form a cell plate. The cell plate then enlarges until its membrane fuses with the plasma membrane of the parent cell. Two daughter cells form. Each has its own plasma membrane. Meanwhile, a new cell wall arising from the content of the cell plate has formed between the daughter cells. Now, let's watch video of cytokinesis in plant cell.
Plant cells, membrane components are assembled in the center of the cell to form the phragmoplast. These components are vesicles derived from the Golgi apparatus. The vesicles travel along microtubules, and their content forms the cell plate. The cell plate continues to grow outward until it reaches the cell membrane along the perimeter of the cell. Cellulose contained in the vesicles is then laid down between the new membranes, creating two new cell walls. Pectin is also deposited in the space between the daughter cells.